So this paper is the key to quasispecies and the quasispecies theory. And you can see it's by the same person that did that review, Raul Andino, and the first author is Marco Vignuzzi. So let's look at this thing. This paper is going to deal with the poliovirus, which causes poliomyelitis in people. And this is a virus that you can also grow in mice, and it's a virus that's been very well studied. And so this is the first paper that I know of that actually directly looked at the consequences of having a quasi-species versus not having a quasi-species. This is kind of the thing that kicks it all off and really gets this thing going. So for starters, let's look at the different kinds of virus that they're going to be using here. So the first one is going to be the wild type virus. And so this thing is going to, they actually sequenced thousands of nucleotides from each one of these things, many genomes, many different individual parts. And this is back at a time when this was very difficult to do, but they still managed to do it. And so what they found is that the wild type has an average of about 1.9, almost 2 mutations per genome. And yeah, great. And then they have, let's see, this one, which is G64S. So this is a mutation in the 3D polymerase of poliovirus. Yeah, you remember 3D, right? And so amino acid 64 should be glycine, but it's turned into serine in this mutant. And that change is something that they figured out actually long ago, changes the polymerase in a way that it still works. It actually works just as fast as normal, but it now is unable to make mistakes. And so the way they selected for this was by putting in chemicals that force the polymerase to make mistakes. So they were basically pushing it off the edge of the error catastrophe cliff. And so the virus responds by coming up with a strain that is able to not make as many mistakes so that it can survive. Yeah. But then when you take away that mutating chemical, then you're left with a virus that just is not very good at making mistakes. So 2 mutations out of 50,000, so about 0.3 mutations per genome. And it's, yeah, about a 6.5 times fewer mutations than the wild type. So they took that one, and then they took that one and grew it, and grew it, and grew it. Because it makes mutations, it just makes them very slowly. And so if you keep it growing and taking it from one cell to another cell to another cell to another cell for a long enough time, you can actually expand it to the point where it'll have about as many mutations in it as the wild type, but they'll all still be on this G64S base. So it won't be able to change quickly, but it'll start out with diversity. And so then this whole paper is going to be about taking these different combinations of virus. So wild type that all start out about the same and make mutations very easily. So they generate a quasi-species when they get to a cell. G64S, you can put in the same amount of this, but they will be almost like clones going into the cell. And so they won't be able to change, but you can put in just as many viruses. And then G64S with an enhanced quasi-species, that's what the EQS is. And that's the one that they grew G64S for long enough that they finally built up these mutations to where it's about like wild type. And then they're going to check it out. So we're going to see what each of these things does inside of a mouse. And so this is a nice short paper. It's something like 3.5 pages of data and a little bit of reference after that, and that's it. So we're going to look at each of the figures. So let's zoom into this figure a little bit. There we go. There we go. Okay, so figure 1. So let's compare. This is mouse survival, and so 100% across the top, this is what you want. This is none of the mice die. If we see these stair steps down, each time we go down, it's one of the mice dying in the experiment. And from this, it looked like they were probably using 10 different mice, I would guess. Because they've got 80%, 50%, 20%, and then 0% still surviving. And so here they're telling you how much virus they put in. So PFU is plaque-forming units. Plaques are a way that we can count viruses. And so this is, think of this as active viruses, a number of active viruses. So for G64S, if you put in 10 to the 7th active viruses, and wild type, you put in 10 to the 7th active viruses, you see how you get a very different curve, right? G64S, you get 0 death, and wild type, you get immediate death within 5 days, but as little as 2 days in every single mouse. So that's like a light switch being on versus off. That is a very stark difference. And since it's the same amount of the same virus, and the only difference is that this one can't make as many mistakes, it actually grows just as fast. That means that the mistake prone virus, is weirdly fitter and more able to cause disease. And in this case, they took this G64S and they put in 10 times as much. You see, with 10 times as much, you can kill a couple mice, but not very many. And with 100 times as much, you can start to kill the mice. It takes several more days. It's like a long, slow process. And even in the end, there was one mouse that stood up to 10 to the 9th active viruses and was basically fine. So this shows that wild type virus is actually really virulent, actually causes a lot of disease, whereas the virus that can't change is not able to cause disease. So let's go over to panel B. This is the one where they compare the wild type virus. So once again, it's the white boxes, and you can see with wild type virus, boom, and we're all dead. Instead of 5 days, one mouse made it to 6 days and then died in this case. That's no big difference. They're comparing that to the same amount, 10 to the 7th, of the G64S with the enhanced quasispecies. So this one starts out as a cloud of viruses. Each one can't change, but individually they start out with a bunch of different ones. And if you look at that, it's pretty much the same thing as the wild type. Maybe it's just like a day slower, but it still kills all the mice. And if you just add 10 times as much of the enhanced quasispecies, then you get basically the exact same curve as for the wild type. Kills them at exactly the same rate. So you can either start out with nothing and make the mutations when you get there. That's what the wild type does. Or you can start out with all the mutations there, and then you don't have to make anything, and that's what the EQS does. Or you can start out with no mutations and not be able to make any mutations, and then the virus is going to lose and the mouse is going to win, and that's what G64S up here is showing you. So that's interesting, right? And we know this for sure applies to poliovirus, but since then there have been some important studies that have shown that a similar sort of thing is probably going on with every RNA virus out there. They're all operating at nearly their error threshold, and they all operate as quasispecies that probably are making you sick because they're able to change while they're inside of you. Yeah, so what's the difference between these things? Well, so let's compare them. So wild type's going to be the square, and the circle is going to be G64S. And so both viruses seem to be able to get into the spleen about as well. G64S isn't quite as good, but similar kinetics. What happens in the muscle? They both get into the muscle, but G64S gets wiped out. Apparently you have to change in some way or make a quasispecies to be able to persist in the muscle. In the spinal cord, you had both of them actually getting in a little bit, but G64S just dies out. It gets shut down, whereas wild type keeps on trucking. Look at that. Kidneys, similar. Small intestines, similar. The brain is just a stark difference. The G64S just doesn't even make it to the brain and doesn't stand a chance. Whereas the wild type, once it gets to the brain, it just keeps mutating and getting better and better and better at growing in the brain. And so shown down here is the difference between the wild type and the G64S, but enhanced quasispecies. So where we start out with the mutations. And if you look, every graph is basically the same because it's able to do everything if it either starts with all the mutations or if it makes the mutations when it's already inside the body. Either way, those are paths to success. But in terms of how the virus is actually spread, you may only have one virus that hits a person. And so if you're a very careful virus like this one, then if only one virus gets in, you're not going to be able to make that quasispecies, and you're not going to survive to actually spread and infect another host. You're going to be extinct, basically. And if you can make mutations like the wild type does, Being able to make these mutations as part of a quasi-species is a survival advantage. So each individual member that has mutations will be weaker than average. But together, because they will have all the right mutations to accomplish the goal, the quasi-species is going to be stronger. And that's the key. That's the key to understanding quasi-species. By having a group of slightly different viruses together, they actually end up being stronger, having a greater effect and a greater survival chance than, a very nearly identical to each other set of viruses, like with G64S. And that's basically it. They then do one more thing where they take virus out of the brain and put it back into the bloodstream, and then go take a look and see, can it make it back to the brain? Because the idea is that, well, what if there are viruses that are just good at getting to the brain? And so they took viruses from wild type and a couple from G64S and G64S EQS that did make it into the muscle, or actually it made it into the brain. And they took these guys out. So these are the ones that have been selected for being able to grow in the brain. And they put them into the bloodstream, and wild type gets right back to the brain. Because wild type doesn't care. It can make any mutations it needs to. But look what happens to the other two. Even if you start with an enhanced quasi-species, if you just pick out any one member, like the best member at getting into the brain, and put it back into the body, it's not going to get to the brain again because they're different skill sets. You need a different set of mutations to be good at getting from the veins into the muscles, a different set of skills to get from the muscles into the nerves, and from the nerves to the spinal cord, and from the spinal cord up to the brain. It just takes different mutations each time, and unless you pack them with you, if you just take one member from this and put them back in, it doesn't work. And so, they're shut down in the spinal cord, and they just don't get to the brain. And that's the idea. And this is them just isolating them back out from the brain and showing that even after all this time, they are all still, all the ones that start out as G64S still have that G64S mutation in there every single time. So there you go. That's the weird, weird world of quasi-species. And that's how viruses work. And you got to really be able to consider this when you're trying to design an antiviral or a therapy, or take care of a person, or look at the way a virus spreads. The virus is never going to stay the same. That's the one thing that it won't do. It's going to evolve as fast as it possibly can because viruses are little evolution machines. All right, I hope you enjoyed that. I hope you got something out of it. Thank you very much.